Resuming on page 59, attachment proteins. Integral proteins may function as attachment proteins, which allow cells to attach to other cells or to extracellular molecules. End of page 59, beginning page 60. Many attachment proteins also attach to intracellular molecules. Cadherins are proteins that attach cells to other cells. Integrins are cells that attach cells to extracellular molecules. Integrins function in pairs of integral proteins which interact with both intracellular and extracellular molecules. Because of the interaction with intracellular molecules, integrins also function in cellular communication. Transport proteins. Transport proteins are integral proteins that allow ions or molecules to move from one side of the plasma membrane to the other. Transport proteins include channel proteins, carrier proteins, and ATP-powered pumps. Pause that reading and read from table 3.2. Functions of membrane proteins. Membrane protein, marker molecule. Protein function allows cells to identify one another or other molecules. Attachment protein anchors cells to other cells, cadherins, or to extracellular molecules, integrins. Transport proteins, channel proteins, form passageways through the plasma membrane, allowing specific ions or molecules to enter or exit the cell, may be gated or non-gated. Carrier proteins, transporters, move ions or molecules across the membrane. Binding of specific chemical to carrier proteins causes changes in the shape of the carrier proteins. The carrier proteins then move the specific chemical across the membrane. ATP-powered pumps move specific ions or molecules across the membrane, require ATP molecules to function. Receptor proteins function as binding sites for chemical signals in the extracellular fluid. Binding of chemical signals to receptors triggers cellular responses. Enzymes catalyze chemical reactions either inside or outside cells. Resuming the main reading. Channel proteins. Channel proteins are one or more integral proteins arranged so that they form a tiny channel through the plasma membrane. The hydrophobic regions of the proteins face outward. End of page 60, beginning page 61. Toward the hydrophobic part of the membrane, and the hydrophilic regions of the protein face inward and line the channel. Ions or small molecules of the right size, charge, and shape can pass through the channel. The charges in the hydrophilic part of the channel proteins determine which type of ions can pass through the channel. Some channel proteins, called non-gated ion channels, are always open and are responsible for the permeability of the plasma membrane to ions when the plasma membrane is at rest. Other channels, called gated ion channels, can be opened or closed. Some gated ion channels open or close in response to chemical signals, or ligands, which are small molecules that bind to the proteins or glycoproteins. These are called ligand-gated ion channels. Other gated ion channels open or close when there is a change in charge across the plasma membrane. These are called voltage-gated ion channels. Carrier proteins. Carrier proteins, or transporters, are integral membrane proteins that move ions or molecules from one side of the plasma membrane to the other. The carrier proteins have specific binding sites to which ions or molecules attach on one side of the plasma membrane. The carrier proteins change shape to move the bound ions or molecules to the other side of the plasma membrane, where they are released. The carrier protein then resumes its original shape and is available to transport more molecules. Movement of ions or molecules by carrier proteins can be classified as uniport, symport, or antiport. Uniport is the movement of one specific ion or molecule across the membrane. Symport is the movement of two different ions or molecules in the same direction across the plasma membrane, whereas antiport is the movement of two different ions or molecules in opposite directions across the plasma membrane. Transport proteins involved in these types of movement are called uniporters, symporters, and antiporters, respectively. ATP-powered pumps. ATP-powered pumps are transport proteins that move specific ions and molecules from one side of the plasma membrane to the other. Unlike carrier proteins, however, the movement of ions or molecules across the membrane is fueled by the breakdown of adenosine triphosphate, ATP. Recall from Chapter 2 that energy stored in ATP molecules is used to power many cellular activities. ATP-powered pumps have binding sites to which specific ions or molecules can bind, as well as a binding site for ATP. The breakdown of ATP to adenosine diphosphate, ADP, releases energy, changing the shape of the protein which moves the ion or molecule across the membrane. Receptor proteins. Receptor proteins are proteins or glycoproteins in the plasma membrane with an exposed receptor site on the outer cell surface, which can attach to specific chemical signals. Many receptors and the chemical signals they bind are part of an intercellular communication system that coordinates cell activities. One cell can release a chemical signal that diffuses to another cell and binds to its receptor. The binding acts as a signal that triggers a response. The same chemical signal would have no effect on other cells that lacked the specific receptor molecule. End of page 61, beginning page 62. Receptors linked to channel proteins. Some membrane-bound receptors also help form ligand-gated ion channels. The ion channels are composed of proteins that span the plasma membrane. Parts of one or more of the channel proteins form receptors on the cell surface. When chemical signals, or ligands, bind to these receptors, the combination alters the three-dimensional structure of the proteins of the ion channels, causing, end of page 62, beginning page 63, the channels either to open or to close. The result is a change in the permeability of the plasma membrane to the specific ions passing through the ion channels. For example, acetylcholine released from nerve cells is a chemical signal that combines with membrane-bound receptors of skeletal muscle cells. The combination of acetylcholine molecules with the receptor sites open sodium ion channels in the plasma membrane. Consequently, the sodium ion diffuse into these skeletal muscle cells and trigger events that cause them to contract. Cystic fibrosis. Cystic fibrosis is a genetic disorder that affects chloride ion channels. There are three types of cystic fibrosis. In about 70% of cases, a defective channel protein is produced that fails to reach the plasma membrane from its site of production inside the cell. In the remaining cases, the channel protein is incorporated into the plasma membrane but does not function normally. In some cases, the channel protein fails to bind ATP. In others, ATP binds to the channel protein, but the channel does not open. Failure of these ion channels to function results in the affected cells producing thick, viscous secretions. Although cystic fibrosis affects many cell types, its most profound effects are in the pancreas and in the lungs. In the pancreas, the thick secretions block the release of digestive enzymes, resulting in an inability to digest certain types of food, and sometimes leading to serious cases of pancreatitis, inflammation of the pancreas. In the lungs, the thick secretions block airways and make breathing difficult. A more detailed description of cystic fibrosis and its consequences can be found in Chapter 23. Receptors linked to G protein complexes. Some membrane-bound receptor molecules function by altering the activity of a G protein complex located on the inner surface of the plasma membrane, which acts as an intermediate between a receptor and other cellular proteins. The G protein complex consists of three proteins called alpha, beta, and gamma proteins. The G protein complex will only associate with a receptor that has a chemical signal bound to it. In its unassociated state, the alpha subunit of the G protein complex has guanosine diphosphate, GDP, attached to it. When a chemical signal binds to the receptor becomes associated with the G protein complex. The alpha subunit releases the GDP and attaches to guanosine triphosphate, GTP. The G protein complex separates from the receptor, and the alpha subunit separates from the beta and gamma subunits. The activated alpha subunit can stimulate a cell response in at least three ways. 1. By means of intracellular chemical signals. 2. By the opening of ion channels in the plasma membrane. And 3. By the activation of enzymes associated with the plasma membrane. Drugs and receptors. Drugs with structures similar to specific chemical signals may compete with those chemical signals for their receptor sites. Depending on the exact characteristics of a drug, it may either bind to a receptor site and activate the receptor, or bind to a receptor site and inhibit the action of the receptor. For example, some drugs compete with the chemical signal for epinephrine for its receptor sites. Some of these drugs activate epinephrine receptors, others inhibit them. Enzymes. Some membrane proteins function as enzymes, which can catalyze chemical reactions on either the inner or the outer surface of the plasma membrane. For example, Some enzymes on the surface, end of page 63, beginning page 64, of cells in the small intestine break the peptide bonds of dipeptides to form two single amino acids. Some membrane-associated enzymes are always active. Others are activated by membrane-bound receptors or G-protein complexes. Movement through the plasma membrane. The plasma membrane separates extracellular material from intracellular material and is selectively permeable. That is, it allows only certain substances to pass through it. The intracellular material has a different composition than the cellular material, and the cell's survival depends on the maintenance of these differences. Enzymes, other proteins, glycogen, and potassium ions are found in higher concentrations intracellularly. Sodium, calcium, and chloride ions are found in greater concentrations extracellularly. End of page 64, beginning page 65. And I'm going to pause just to make a note. There are many illustrations and graphics in this book and charts and such with notes, but I'm not going to post a photo of them and show them because of copyright. And there's a lot of notes with them, so I may pick and choose what I read from those illustrations, but I intend to read the tables and side texts. But if I just read the notes for different pictures without showing it, people may make sense of it, people may not, but that is all. I'm going to resume page 65. In addition, nutrients must continually enter the cell and waste products must exit, but the volume of the cell remains unchanged. Because of the plasma membrane's permeability characteristics and its ability to transport molecules selectively, the cell is able to maintain homeostasis. Rupture of the membrane, alteration of its permeability characteristics, or inhibition of transport processes can disrupt the normal concentration differences across the plasma membrane and lead to cell death. Molecules and ions can pass through the plasma membrane in four ways. 1. Directly. Directly through the phospholipid membrane. Molecules that are soluble in lipids, such as oxygen, carbon dioxide, and steroids, pass through the plasma membrane readily by dissolving in the lipid bilayer. The phospholipid bilayer acts as a barrier to most substances that are not lipid soluble, but some small non-lipid soluble molecules such as carbon dioxide and urea can diffuse between phospholipid molecules of the plasma membrane. 2. Membrane channels. There are several types of protein channels through the plasma membrane. Each channel type allows only certain molecules to pass through it. The size, shape, and charge of molecules determine whether they can pass through a given channel. For example, sodium ions pass through sodium channels, and potassium and chloride ions pass through potassium and chloride channels, respectively. Rapid movements of water across the cell membrane also occurs through membrane channels. 3. Transport proteins. Large polar molecules that are not lipid soluble, such as glucose and amino acids, cannot pass through the plasma membrane in significant amounts unless they are moved across by transport proteins. Substances that are moved across the plasma membrane by transport proteins are said to be transported by mediated processes. Specific molecules bind to specific transport proteins that carry them across the plasma membrane. Transport proteins that move glucose across the plasma membrane do not move amino acids, and transport proteins that move amino acids across the plasma membrane do not move glucose. 4. Vesicles. Large non-lipid soluble molecules, small pieces of matter, and even whole cells can be transported across plasma membrane in a vesicle, which is a small sac surrounded by a membrane. Because of the fluid nature of membranes, the vesicle and the plasma membrane can fuse, allowing the contents of the vesicle to cross the plasma membrane. Diffusion. A solution consists of one or more substances called solutes, dissolved in the predominant liquid or gas, which is called the solvent. Diffusion is the movement of solutes from an area of higher solute concentration to an area of lower solute concentration. Diffusion is a product of the constant random motion of all atoms, molecules, or ions in a solution. Because there are more solute particles in an area of higher concentration than in an area of lower concentration, and because the particles move randomly, the chances are greater that solute particles will move from the higher to the lower concentration than in the opposite direction. Thus, overall or net movement is from the area of higher solute concentration to that of lower solute concentration. At equilibrium, the net movement of solutes stops, although the random molecular motion continues, and the movement of solutes in any one direction, is balanced by an equal movement in the opposite direction. The movement and distribution of smoke or perfume throughout a room without air currents, and of a dye throughout a beaker of still water are examples of diffusion. A concentration difference occurs when the concentration of a solute is greater at one point than at another point in a solvent. The concentration difference between 2 points divided by the distance, end of page 65, beginning page 66, between the 2 points is called the concentration or density gradient. I go back to page 65 and read some notes of an illustration. So there's 3 images of a beaker. There's distilled water, and then there's a block of salt, or so concentration gradient for salt, distilled water. So 1, when a salt crystal is placed into a beaker of water, there is a concentration gradient for salt from the salt crystal to the water that surrounds it. There is also a concentration gradient for water molecules from the water toward the salt crystal. 2. Salt ions move down their concentration gradient from an area of high concentration to an area of low concentration into the water. And as a note, basically it's just describing the block breaking down and spreading to different areas of the water-filled beaker. Resuming 3. Salt ions and water molecules are distributed evenly throughout the solution. Even though the salt ions and water molecules continue to move randomly, An equilibrium exists and no net movement occurs because no concentration gradient exists. Returning to page 67 to resume the main reading. Solutes diffuse down their concentration gradients from a higher to a lower solute concentration until an equilibrium is achieved. The greater the concentration gradient, the greater the rate of diffusion of a solute down that gradient. Increasing the concentration difference or decreasing the distance between the 2 points increases the concentration gradient, whereas the concentration difference or increasing the distance between the 2 points decreases the concentration gradient. The rate of diffusion is influenced by the magnitude of the concentration gradient, the temperature of the solution, the size of the diffusing molecules, and the viscosity of the solvent. The greater the concentration gradient, the greater the number of solute particles moving from a higher to a lower solute concentration. As the temperature of a solution increases, the speed at which all molecules move increases, resulting in a greater diffusion rate. Small molecules diffuse through a solution more readily than do large ones. Viscosity is a measure of how easily a liquid flows. Thick solutions, such as syrup, are more viscous than water. Diffusion occurs more slowly in viscous solvents than in thin, watery solvents. Diffusion of molecules is an important means by which substances move between the extracellular and intracellular fluids in the body. Substances that can diffuse through either the lipid bilayer or the membrane channels can pass through the plasma membrane. Some nutrients enter and some waste products leave the cell by diffusion, and maintenance of the appropriate intracellular concentration of these substances depends to a large degree on diffusion. For example, if the extracellular concentration of oxygen is reduced, inadequate oxygen diffuses into the cell, and normal cell function cannot occur. Some lipid-soluble chemical signals can diffuse through the plasma membrane and attach to receptors inside the cell. Urea is, I'm going to read a note that says Predict 1. Urea is a toxic waste produced inside cells. It diffuses from the cells into the blood and is eliminated from the body by the kidneys. And then it asks, what would happen to the intracellular and extracellular concentration of urea if the kidneys stopped functioning? And then we move on to the next section. Osmosis. Osmosis is the diffusion of water, solvent, across a selective permeable membrane, such as a plasma membrane. A selectively permeable membrane is a membrane that allows water, but not all the solutes dissolved in the water, to diffuse through the membrane. Aquaporins, or water channel proteins, increase membrane permeability to water in some cell types, such as kidney cells. Water diffuses from a solution with proportionately more water across a selectively permeable membrane and into a solution with proportionately less water. Because solution concentrations are defined in terms of solute concentrations, not in terms of water content, see chapter 2, water diffuses from the less concentrated solution, fewer solutes, more water, into the more concentrated solution, more solutes, less water. Osmosis is important to cells because large volume changes caused by water movement disrupt normal cell function. Osmotic pressure is the force required to prevent the movement of water by osmosis across a selectively permeable membrane. End of page 66, beginning page 67. The osmotic pressure of a solution can be determined by placing the solution into a tube that is closed at one end by a selectively permeable membrane. The tube is then immersed in a distilled water. Water molecules move by osmosis through the membrane into the tube, forcing the solution to move up the tube. As the solution rises into the tube, its weight produces hydrostatic pressure, which moves water out of the tube back into the distilled water surrounding the tube. At equilibrium, net movement of water stops, which means the movement of water into the tube by osmosis is equal to the movement of water out of the tube caused by hydrostatic pressure. The osmotic pressure of the solution in the tube is equal to the hydrostatic pressure that prevents net movement of water into the tube. The osmotic pressure of a solution provides information about the tendency for water to move by osmosis across a selectively permeable membrane. Because water moves from less concentrated solutions, fewer solutes, more water, into more concentrated solutions, more solutes, less water. The end of page 67, beginning page 68. Greater the concentration of a solution, the less water it has. The greater the tendency for water to move into the solution, and the greater the osmotic pressure to prevent that movement. Thus, the greater the concentration of a solution, the greater the osmotic pressure of the solution, and the greater the tendency for the water to move into the solution. Three terms describe the osmotic pressure of solutions. Solutions with the same concentration of solute particles, see chapter 2, have the same osmotic pressure and are referred to as isosmotic. The solutions are still isosmotic even if the types of the solute particles in the two solutions differ from each other. If one solution has a greater concentration of solute particles and therefore a greater osmotic pressure than another solution, the first solution is said to be hyperosmotic, compared with the more dilute solution. The more dilute solution with the lower osmotic pressure is hyposmotic, compared with the more concentrated solution. Three additional terms describe the tendency of cells to shrink or swell when placed into a solution. If a cell is placed into a solution in which it neither shrinks nor swells, the solution is said to be isotonic. In an isotonic solution, the shape of the cell remains constant, maintaining its internal tension or tone, a condition called tonicity. If a cell is placed into a solution and the water moves out of the cell by osmosis, causing the cell to shrink, the solution is called hypertonic. If a cell is placed into a solution and water moves into the cell by osmosis, causing the cell to swell, the solution is called hypotonic. An isotonic solutions may be isosmotic to the cytoplasm because isosmotic solutions have the same concentration of solutes and water as the cytoplasm of the cell. No net movement of water occurs, and the cell neither swells nor shrinks. Hypertonic solutions can be hyperosmotic and have a greater concentration of solute molecules and a lower concentration of water than the cytoplasm of the cell. Therefore, water moves by osmosis from the cell into the hypertonic solution, causing the cell to shrink, a process called crenation in red blood cells. Hypotonic solutions can be hyposmotic and have a smaller concentration of solute molecules and a greater concentration of water than the cytoplasm of the cell. Therefore, water moves by osmosis into the cell, causing it to swell. If the cell swells enough, it can rupture, a process called lysis. Solutions injected into the circulatory system or the tissues must be isotonic because shrinkage or swelling of cells disrupts their normal function and can lead to cell death. The osmotic terms refer to the concentration of the solutions, and the tonic terms refer to the tendency of cells to swell or shrink. These terms should not be used interchangeably. Not all isosmotic solutions are isotonic. For example, it is possible to prepare a solution of glycerol and a solution of mannitol that are isosmotic to the cytoplasm of the cell. Because the solutions are isosmotic, they have the same concentration of solutes and end of page 68, beginning page 69, water as the cytoplasm. Glycerol, however, can diffuse across the plasma membrane, but mannitol cannot. When glycerol diffuses into the cell, the solute concentration of the cytoplasm increases, and its water concentration decreases. Therefore, water moves by osmosis into the cell, causing it to swell, and the glycerol solution is both isosmotic and hypertonic. In contrast, mannitol cannot enter the cell, and the isosmotic mannitol solution is also isotonic. Filtration. Filtration results when a partition containing small holes is placed in a stream of moving liquid. The partition works as a minute sieve. Particles small enough to pass through the holes move through the partition with the liquid, but particles larger than the holes are prevented from moving beyond the partition. In contrast to diffusion, filtration depends on a pressure difference on either side of the partition. The liquid moves from the side of the partition with the greater pressure to the side with the lower pressure. Filtration occurs in the kidneys as a step in urine formation. Blood pressure moves fluid from the blood through a partition or filtration membrane. Water, ions, and small molecules pass through the partition, whereas most proteins and blood cells remain in the blood. Mediated transport. Many essential molecules, such as amino acids and glucose, cannot enter the cell by diffusion, and many products, such as proteins, cannot exit the cell by diffusion. Mediated transport is the process by which transport proteins mediate or assist in the movement of large water-soluble molecules or electrically charged molecules or ions across the plasma membrane. All three types of transport proteins, carrier proteins, transporters, ATP-powered pumps, and channel proteins, ion channels, are involved in mediated transport. Mediated transport has three characteristics: specificity, competition, and saturation. Specificity means that each transport protein binds to and transports only a single type of molecule or ion. For example, the transport protein that moves glucose does not move amino acids or ions. The chemical structure of the binding site determines the specificity of the transport protein. Competition is the result of similar molecules binding to the transport protein. Although the binding sites of transport proteins exhibit specificity, closely related substances, in which regions of two different molecules have the same shape, may bind to the same binding site. The substance in the greater concentration, or the substance that binds to the binding site more readily, is moved across the plasma membrane at the greater rate. Saturation means that the rate of movement of molecules across the membrane is limited by the number of available transport proteins. As the concentration of a transported substance increases, more transport proteins have their binding sites occupied. The rate at which the substance is moved across the plasma membrane increases. However, once the concentration of the substance is increased so that all the binding sites are occupied, the rate of movement remains constant, even though the concentration of the substance increases further. Ion channels often are thought of as simple tubes, with or without gates, through which ions pass. Many ion channels, however, appear to be more complex than once thought. It now appears that ions briefly bind to specific sites inside channels, and that there is a change in the shape of those channels as ions are transported through them. The size and charge within a channel determine the channel's specificity. For example, sodium ion channels do not transport potassium ions and vice versa. In addition, similar ions moving into and binding within an ion channel are in competition with each other. Furthermore, the number of ions moving into an ion, end of page 69, beginning page 70, channel can exceed the capacity of the channel, thus saturating the channel. Therefore, ion channels exhibit specificity, competition, and saturation. Three kinds of mediated transport exist: facilitated diffusion, active transport, and secondary active transport. Facilitated diffusion. Facilitated diffusion is a carrier-mediated or channel-mediated process that moves substances into or out of cells from a higher to a lower concentration. Facilitated diffusion does not require metabolic energy to transport substances across the plasma membrane. The rate at which molecules or ions are transported is directly proportional to their concentration gradient up to the point of saturation, when all the carrier proteins or channels are occupied. Then the rate of transport remains constant at its maximum rate. Active transport. Active transport is a mediated transport process that requires energy provided by ATP. movement of the transported substance to the opposite side of the membrane and its subsequent release from the ATP-powered pump are fueled by the breakdown of ATP. The maximum rate at which active transport proceeds depends on the number of ATP-powered pumps in the plasma membrane and the availability of adequate ATP. Active transport processes are important because they can move, end of page 70, beginning page 71, substances against their concentration gradients, that is, from lower concentrations to higher concentrations. Consequently, they can accumulate substances on one side of the plasma membrane at concentrations many times greater than those on the other side. Active transport can also move substances from higher to lower concentrations. Some active transport mechanisms exchange one substance for another. For example, the sodium-potassium Na+-K+ pump moves sodium out of cells and potassium into cells. The result is a higher concentration of sodium outside the cell and a higher concentration of potassium inside the cell. Because ATP is broken down during the transport of sodium and potassium, the pump is also called sodium-potassium ATPase. The sodium-potassium pump is very important to a number of cell functions. These are discussed in chapters 9 and 11. Secondary active transport. Secondary active transport involves the active transport of an ion, such as sodium, out of a cell, establishing a concentration gradient with a higher concentration of the ions outside the cell. The tendency for the ions to move back into the cell down their concentration gradient provides the energy necessary to move a different ion or some other molecule into the cell. For example, glucose is moved from the lumen of the intestine into the epithelial cells by secondary active transport. This process requires two transport proteins. A sodium-potassium pump actively moves sodium out of the cell, and two, a carrier protein facilitates the movement of sodium and glucose into the cell. Both sodium and glucose are necessary for the carrier protein to function. The movement of sodium down their concentration gradient provides the energy to move glucose molecules into the cell against their concentration gradient. Thus, glucose can accumulate at concentrations higher inside this cell than outside. Because the movement of glucose molecules against the concentration gradient results from the formation of a concentration gradient of sodium by an active transport mechanism, the process is called secondary active transport. Endocytosis and exocytosis. Endocytosis, or the internalization of substances, includes both phagocytosis and pinocytosis, and refers to the uptake of material through the plasma membrane by the formation of a vesicle. A vesicle is a membrane-bound sac found within the cytoplasm of a cell. A portion of the plasma membrane wraps around a particle or droplet and fuses so that the particle or droplet is surrounded by a membrane. That portion of the membrane then, quote, pinches off, end quote, so that the particle or droplet surrounded by a membrane is within the cytoplasm of the cell, and the plasma membrane is left intact. Phagocytosis literally means cell eating and applies to endocytosis, when solid particles are ingested and phagocytic vesicles are formed. White blood cells and some other cell types phagocytize bacteria, cell debris, and foreign. End of page 71, beginning page 72. Particles. Phagocytosis is therefore important in the elimination of harmful substances from the body. Pinocytosis means cell drinking and is distinguished from phagocytosis in that smaller vesicles are formed and they contain molecules dissolved in liquid rather than particles. Pinocytosis often forms vesicles near the tips of deep invaginations of the plasma membrane. It is a common transport phenomenon in a variety of cell types and occurs in certain cells of the kidneys, epithelial cells of the intestines, cells of the liver, and cells that line capillaries. Endocytosis can exhibit specificity. For example, cells that phagocytize bacteria and necrotic tissue do not phagocytize healthy cells. The plasma membrane may contain specific receptor molecules that recognize certain substances and allow them to be. End of page 72, beginning page 73. Transported into the cell by phagocytosis or pinocytosis. This is called receptor-mediated endocytosis, and the receptor sites combine only with certain molecules. This mechanism increases the rate at which specific substances are taken up by the cells. Cholesterol and growth factors are examples of molecules that can be taken into a cell by receptor-mediated endocytosis. Both phagocytosis and pinocytosis require energy in the form of ATP and therefore are active processes. Because they involve the bulk movement of material into the cell, however, phagocytosis and pinocytosis do not exhibit the degree of specificity or saturation that active transport exhibits. End of page 73, beginning page 74. Hypercholesterolemia. Hypercholesterolemia is a common genetic disorder affecting 1 in every 500 adults in the United States. It consists of a reduction in or an absence of low-density lipoprotein, LDL, receptors on cell surfaces. This interferes with receptor-mediated endocytosis of LDL cholesterol. As a result of inadequate cholesterol uptake, cholesterol synthesis within these cells is not regulated, and too much cholesterol is produced. The excess cholesterol accumulates in blood vessels, resulting in atherosclerosis. Atherosclerosis can result in heart attacks or strokes. A more detailed description of hypercholesterolemia can be found in chapter 24. Resuming the main reading. In some cells, secretion can accumulate within vesicles. These secretory vesicles then move to the plasma membrane, where the membrane of the vesicle fuses with the plasma membrane and the content of the vesicle is expelled from the cell. This process is called exocytosis. Secretion of digestive enzymes by the pancreas and secretion of mucus by the salivary glands are examples of exocytosis. Table 3.3 summarizes and compares the mechanisms by which different kinds of molecules are transported across the plasma membrane. End of page 74, beginning page 75. Table 3.3, comparison of membrane transport mechanisms. Transport mechanism, diffusion. Description. Random movement of molecules result in net movement from areas of higher to lower concentration. Substance transporter. Lipid-soluble molecules dissolve in the lipid bilayer and diffuse through it. Ions and small molecules diffuse through membrane channels. Example: oxygen, carbon dioxide, and lipids, such as steroid hormones, dissolve in the lipid bilayer. Chloride ions and urea move through membrane channels. Transport mechanism, osmosis. Description. Water diffuses across selectively permeable membrane. Substances transported. Water diffuses through the lipid bilayer. Example: water moves from the intestines into the blood. Filtration. Liquid moves through a partition that allows some, but not all, of the substances in the liquid to pass through it. The movement is due to a pressure difference across the partition. Liquid and substances pass through holes in the partition. Filtration in the kidneys allows removal of everything from the blood except proteins and blood cells. Facilitated diffusion. Carrier proteins combine with substances and move them across the plasma membrane. No ATP is used. Substances are always moved from areas of higher to lower concentration. It exhibits the characteristics of specificity, saturation, and competition. Some substances too large to pass through membrane channels and too polar to dissolve in the lipid bilayer are transported. Glucose moves by facilitated diffusion into muscle cells and fat cells. Active transport. ATP-powered pumps combine with substances and move them across the plasma membrane. ATP is used. Substances can be moved from areas of lower to higher concentration. It exhibits the characteristics of specificity, saturation, and competition. Substances too large to pass through channels and too polar to dissolve in the lipid bilayer are transported. Substances that are accumulated in concentrations higher on one side of the membrane than on the other are transported. Ions such as sodium, potassium, and calcium cation or calcium ion are actively transported. Secondary active transport. Ions are moved across the plasma membrane by active transport, which establishes an ion concentration gradient. ATP is required. Ions then move back down their concentration gradient by facilitated diffusion, and another ion or molecule moves with the diffusion ion, symport, or in the opposite direction, antiport. Some sugars, amino acids, and ions are transported. There is a concentration gradient for sodium into intestinal epithelial cells. This gradient provides the energy for the symport of glucose. As sodium enters the cell, down their concentration gradient, glucose also enters the cell. In many cells, hydrogen ions are moved in the opposite direction of sodium, antiport. Endocytosis. The plasma membrane forms a vesicle around the substances to be transported, and the vesicle is taken into the cell. This requires ATP in receptor-mediated endocytosis. Specific substances are ingested. Phagocytosis takes in cells and solid particles. Pinocytosis takes in molecules dissolved in liquid. Immune system cells, called phagocytes, ingest bacteria and cellular debris. Most cells take in substances through pinocytosis. Exocytosis. Materials manufactured by the cell are packaged in secretory vesicles that fuse with the plasma membrane and release their contents to the outside of the cell. This requires ATP. Protein and other water-soluble molecules are transported out of cells. Digestive enzymes, hormones, neurotransmitters, and glandular secretions are transported, and waste products are eliminated. And cell waste products are eliminated. End of page 75, beginning page 76.